Good morning, good afternoon, and good evening, everyone. This is Liam Sanyo from Inside Scientific, and I'm pleased to be your host and moderator for today's event, which is titled The Wonderful World of Scanning Electrochemical Microscopy. Today, we're very fortunate to be joined by Janine Mauzeroll, Professor of Chemistry at McGill University and Head of the Laboratory for Electrochemical Reactive Imaging and Detection of Biological Systems. Today, Professor Mauzeroll will discuss the fundamentals, critical experimental parameters, and recent applications for scanning electrochemical microscopy, or SECM for short. And with that, I'm very pleased to introduce the speaker, Dr. Janine Mauzeroll. Janine, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Thank you very much. Hi, everybody. My name is Janine. Thanks for answering the poll because it's really useful for me to gauge the level of the talk. And I'd like to let you know that there's a question and answer chat. So if you do have specific questions during the talk, please put them there. I'll be answering all the questions that I have tomorrow. And so you'll be sure to have a response. So please don't be shy if you have any questions at all. So, thank you, Liam, for the kind invitation and for organizing the webinar, Jamie, Frank, and Mareke. It's really appreciated. So let's get started. And I always start by acknowledgements because, as many people know, none of the great science that we do actually occurs without a wonderful team. And so I'd like to acknowledge all my current lab members, which are on top, and my past group members that have in some way contributed to some of the topics that we'll be discussing today. Without them, nothing would be happening in our group. So today is all about scanning electrochemical microscopy. And we see on the left-hand side in blue a micropipette tip who's basically playing ping pong with a surface using a redox probe highlighted in red and green. And so this redox probe in scanning electrochemical microscopy gets oxidized or reduced at the surface and then diffuses back to the microelectrode, where it will exchange electrons and generate a current, which we will be monitoring. So on this slide, I'm trying to showcase that there are different types of modes of SECM. If we look in panel A, on the top, you have the microelectrode and you see R and O, which are our redox species in solution. The R demonstrate that there is a hemispherical diffusion of the species to the electrode surface. And so therefore, scanning electrochemical microscopy is highly influenced by how fast species move in a particular media, but also how fast that electron, which you see in the dark portion of the microelectrode, gets transferred. And so when the microelectrode is very far away from solution, we have hemispherical diffusion. And when we decide to approach the microelectrode to a surface, which is highlighted in gray, we see that the diffusion of the species to the microelectrode surface is sometimes hindered in panel B by the physical presence of the substrate, or in C, it can be influenced by how fast that electron transfer occurs between species O and R at the substrate, which is in gray. And we can see that you can also collect a species that's formed at a surface in D. You can compete with the consumption of a species in solution, which is called redox competition mode in F. And you have modes also that don't necessarily monitor the current. As I said, you have modes that involve the use of ion selective electrodes, which is presented in panel H as the potentiometric mode. So the point of this slide is to show you that in fact SECM is quite versatile and you can apply it to a wide variety of situations where your substrate is reacting or generating a species of interest. Now, for those of us who love instruments, let me just briefly mention what the instrumental setup of an SECM looks like. At the bottom center, you see that we have a working electrode, a microelectrode, which is where all the action is going to occur. We have a reference electrode and a counter electrode, and a second working electrode, which oftentimes is the substrate. Although it could be connected to a bipotentiostat, and actually we can use the bipotentiostat to apply a potential or a current at the substrate, it doesn't necessarily need to be poised for SECM to work. So that's the electrochemical part of the system. And then one of the key features of scanning electrochemical microscopy is the fact that you can acquire reactive imaging of the surface by rastering that microelectrode across the surface using high positioning motors. Now, in some systems, you can also have additional distance controllers, like a shear force controller, for example, that consistently monitors the tip to substrate distance if your objective is to perform a measurement at a constant distance. And there are several different types of ways by which you can keep a constant distance. Many people are actually professionals at building their own instruments. So I'm just trying to provide you with one example of how an SECM microscope works. So fundamentally, my SECM is monitoring the ping pong match between the redox mediator in solution and the surface, as you see in the animation on the left. And what we're measuring is the current, and that is related to the rate of the electrochemical reaction that's occurring in solution. And so if we think about it a little bit more, by controlling the applied potential either at the microelectrode or at the underlying substrate, what we're effectively doing is controlling the delta G of the reaction using the applied potential, making it more or less favorable, which is what I'm trying to demonstrate here in the red curve that now has a more favorable delta G in order for the reaction to occur. So what do we need to know to quantify the current that we are going to be measuring at the microelectrode? Well, we need to know two things. We need to know what is the rate of electron transfer at the microelectrode and at the substrate, and the rate of mass transport, how fast that red and blue ball are actually taking to diffuse or migrate in between this cleft. In order to understand this, many researchers that use SECM will rely on numerical simulation when they want to understand something about the heterogeneous electron transfer rate that's occurring at the working electrode. They'll be using the Butler-Volmer relationship, which you have on slide 14, and which is a classical expression in electrochemistry. So they'll use this expression in order to understand the electron transfer rate that's occurring. And then to understand how species are moving in solution, well, they'll be using relationships like the Nernst-Planck mass transport. And here you see on the right-hand side that the electrolyte is green, highlighting that the species that are moving in this electrolyte, in this case, either through diffusion or through migration. So in order to have a quantitative understanding of the current, we need to combine these expressions related to the electrokinetics and related to the mass transport in the electrolyte. And the tool that many people use is based on finite element modeling. So in panel 1 on the top left-hand side, I'm showing you a typical geometry that we use to start off our simulations. You see that you have the microelectrode on top. You have a species that's undergoing an electrochemical process from O to R at a certain rate. You have the electrolyte, in which you will have concentration profiles. And you have the substrate in gray that will also have a certain rate at which the reaction R2O is going to occur. Once you've identified which boundaries will have a mass transport component or will be subject to electrode kinetics, then you go in panel 2 where you will mesh that in solution environment. So you will define small boxes that can be triangular or rectangular in geometry, and you will solve the equations discretely in these elements. Once you've solved them, then we see on panel 3 on the lower left-hand side, you can obtain concentration profiles close to the electrode surface or close to the substrate if you would like. Once you have access to these concentration profiles, you can then reproduce the approach curves, which are presented in panel 4 on the lower right-hand side. An approach curve is a type of SECM measurement that we carry out when we approach the microelectrode in a direction normal to the surface that we're trying to study. So that's why on the y-axis, you have the normalized current. That's the current measured at the microelectrode with respect to the current that you would have measured when the microelectrode is in the bulk solution, very far away from the substrate. And on the x-axis, you have the normalized distance, which represents the tip to substrate distance, and it's normalized by typically the radius of the microelectrode that you use. So you see that at a distance of 3, we are much further away from the surface. And as we approach the microelectrode towards 1 and 0, we see contrast in the current that we measure. And that's what I want to discuss with you a little bit further. But the point of this slide is to tell you that if your objective is to quantify kinetics or the reactivity of your underlying surface, it's usually not enough in SECM to just carry out the experiment. You also have to develop models in order to fit approach curves to extract the parameters. So here we have a summary slide where you have the normalized electrode current, NIT, and that normalized current, which we will be simulating or measuring experimentally, is going to be a function of several parameters, including L, that's that tip to substrate distance, which you see on the lower left-hand side. RG is an important parameter in SECM. It's the normalized ratio between the RG of the insulating sheet and the radius of the active portion of that microelectrode. And so this is where we see that being able to characterize and know what the RG of the probes that you are using, that's really important if your objective is to do quantitative SECM, because you will need to include it in your model. And kappa, that's related to the kinetics of the underlying surface. So if we want to recap the behavior that we can observe during typical SECM experiments, if we approach a microelectrode to an insulating surface, piece of plastic, um, a piece of glass, a surface that's covered with a non-conductive protein, then as the tip to substrate distance decreases, we will progressively measure a decrease in the current at our microelectrode because the insulator is physically blocking the hemispherical diffusion of the species in solution. And that we call true negative feedback, where the response is really governed by hindered diffusion and physical presence of the substrate. If we have a conductor, a piece of gold, a piece of platinum, a piece of carbon that's well poised, when we decrease the tip to substrate distance, yes, we will still have hindered diffusion by the physical presence of the conductor, but it will be outweighed by the additional flux that's coming because of the reaction at the conductor surface, which is why as we decrease the tip to substrate distance, the red curve becomes progressively higher. And that's what we call positive and negative feedback. This is the most contrast that you can think to have in SECM. Now, the imaging component of SECM comes in when you choose to select a certain tip to substrate distance highlighted in the dashed line, and then raster that microelectrode across the XY plane of your substrate. And when you do this, it's possible for you to pick up contrast in terms of reactivity of the surface. And then you end up having SECM images across the XY plane, where in the middle, for example, I would have a hot feature where the current would be much higher in red, around 100 picoamps. And if it's surrounded by an insulator, the current would be much lower. One thing you can notice is that SECM images will never be able to reach the lateral resolution that you can expect to have using other scanning probe methods like AFM or STM. The process that's going on here is based on diffusion, which in a way is blurring the reactivity of the feature. So the advantage of SECM is that you can directly measure the flux, but the lateral resolution, unless you decide to combine your SECM with an AFM, which there are several groups in the world that are really good at doing this, it will be hard to have a lateral resolution with normal commercial instruments that can compete with AFM and STM. And so we have in green and red our feedback contrast. And if our underlying surface has intermediate kinetics, then we will get a family of curves in blue. And this is when we would want to fit the experimental approach curves and try to extract the kinetics of the underlying surface. So from this discussion, one key thing that comes out is that microelectrodes are really important. And knowing the geometry of your electrode and being able to reproducibly produce microelectrodes is key. There are many types of probes. You see on the top part of the slide in A, B, C, E, that you can have a disc geometry electrode. In this case, it's a platinum. You can have nanometer sized electrodes, which are obtained using a laser pulling procedure. You can have hemispherical electrodes. In this case, it was the mercury deposited hemisphere in panel C. And you can also have ring electrodes that are built around classical electrophysiology type pipettes. Also, as SECM has progressed, multifunctional probes have started to emerge, being able to combine the two different detection scheme, And you see on the combination of AFM SECM, the use of dual platinum and carbon probes. And so there's a lot of work and research in SECM related to the fabrication of the probes. And there are very few of them that are commercially available. Typically, the ones that are commercially available will be the ones that you see in the top left-hand side panel in A. Disc type electrodes are usually commercially available, and their size are between 25 micron all the way down to 1 micron, but very few commercial probes are sold below these types of dimensions. If we look at the application field of SECM, we see in black that the kinetics of surfaces has been really well studied. In pale blue, biology has been studied to a great extent. In dark blue, we have instrumental development, the hyphenation of SECM with AFM, with the fluorescence microscopy, with Raman. It's really ongoing. Corrosion field has been using the SECM. Now, progressively, more and more, the energy field as well, and all manufacturing type of surface modification can also benefit from SECM in terms of identifying porosity or defects in surface coatings. So we're now pretty much like halfway through the webinar. This was the portion where I was trying to present some of the principles. If you have questions, please let me know. So now I'll try to showcase 3 short stories to demonstrate the application field and the type of research you could do. I won't go in great details. It's just to give you an idea of how you could use these methods, maybe perhaps in your work. So in our group, one thing that really interested us was to use SECM to monitor the flux of species coming from cancer cells. Now, if you, on the left-hand side, you have a normal cell. In this case, for this work, we were using HeLa cells. And on the cell membrane of all cells, you have transmembrane proteins that are there to detoxify your body, and they're highlighted in purple. Now, when a person has cancer and is exposed to large amounts of chemotherapeutics for a large time, here highlighted in green, what happens is that the expression level of these transmembrane pumps in purple is increased. And you develop the phenotype of multi-drug resistance, which means that the chemotherapeutic agent is more easily exported out of the cell and therefore less likely to have the desired effect. So we wanted to see if SECM could be used to monitor the functional activity of these transmembrane proteins. And so we drug challenged the cells. We drug challenged cells that were not resistant on the left-hand side and cells that were resistant. So we generated a population of cells that had different MDR phenotype. Next, as we discussed, SECM is a surface-based technique, and so many people that work with live cells rely on a soft type lithography strategy, where they will be able to pattern their cells. In this case, I'm showing you the protocol that we use in our labs, but there are people that also use conical vials or conical miniaturized dishes. And so there's a lot of activity in this field as well. So you pattern the cells. And then what we did is we used 2 redox mediators to acquire images. On the left-hand side in panel A, the orange ball is ferrocene methanol, which once oxidized to the ferrocenium ion can react with glutathione, which is the green ball that's being exported out of this transmembrane pump. And so here you're going to have an SECM response that depends on the functional activity of that MRP1 pump. Now, cells have a huge topography. And so if you don't have a distance controller like a shear force controller, you need an alternate scheme to decouple the response, the functional response of the pump that you're trying to look at from the overall topography of the cell. And so using the pink ball, the hexaamine ruthenium chloride reduction at the microelectrode, generates species that are highly charged. And as long as your cell is living and that the cell membrane integrity is not compromised, then you're able to use this probe as a topographical tool. And by subtracting the topography from the reactivity, you're basically carrying out two images, one after the other. One in ferrocene methanol, then you change the liquid and you do one in hexaamine ruthenium chloride. You're then able to extract a kind of a kinetic map. And then you can imagine changing many different types of experimental parameters. In this case, I'm showing you how the activity of the cell was affected by the pattern size. In pattern size that are not well suited, where the cell cannot adhere properly, like in the ones for 30 and 50 microns, you see large red areas, meaning that the cancer cells are highly stressed and unhappy. You can carry out time-lapse imaging by exposing your cancer cells to different chemical species and then monitoring their oxidative stress or their response or their cell death as well. And you can also look at the different cell populations that I was mentioning that were particularly targeting MRP1 phenotype and get an idea of how much more active certain cells are and try to relate the functional activity that you measure in SECM with the expression level. So that's what that was the example for cancer. Let me now switch gears and talk to you a little bit about how SECM can be used for batteries. And in this case, we will slightly change the experimental setup that we've been discussing up to now. There are many ways by which SECM can be used to study energy materials. You can apply them to entire cathode or anode films. You can evaluate the effect of film porosity using SECM, but you can also answer questions like, are all battery particles created equal? And that's because of the high resolution capabilities of the method. So in this case, here's the experimental setup if you want to look at very small battery material aggregates. Rather than using the classical microelectrode that we discussed in the previous story, here would be using a hollow micropipette, which is filled with an electrolyte. Now, if you want to do battery material, this type of electrolyte in the work that I'll be showing you is an organic electrolyte like propylene carbonate. And then you can form a small droplet at the end of this micropipette and basically isolate single battery particles or aggregates and look at intercalation reactions, for example. So the principle behind the method is that you have this hanging drop at the end of your pipette and you bring it on a surface, carry out your electrochemical process, and then you can move it along the surface. And with the SECM, by monitoring the current, you can set a threshold at which will help you to land the micropipette on the surface. And this is the second panel on the right-hand side. Once you land, you can carry out a voltammogram, a galvanostatic experiment, coulometry, whatever you would normally be able to carry out with a potentiostat. So This is really interesting because you control very precisely the time of exposition of your surface to the electrolyte. It's not that critical for the battery work that I'm discussing here, but it's definitely important in terms of corrosion studies, which we'll talk about later on. So it allows you to perform large matrix scans over millimeter scale, and acquire an enormous amount of data. And in this case, because it was a battery work, of course, the entire system had to be put under an anaerobic condition in a glove box. And it's pretty doable. You see on the left-hand side the glove box, and in the zoomed in area, you can see the small micropipette and the substrate where the battery particle were basically dispersed. I would say it's the hardest part of this type of experiment is learning to work with such delicate electrodes using the glove box gloves. But there's very few experiments you can carry out with an aqueous electrolytes for batteries. So if your field is battery research and you're thinking about doing such measurements, you have to think about putting the entire system in a glove box. So here I'm just putting up again our scheme where we have, in this case, the battery particle that we're going to be looking at is going to be LFP. In black, we're going to be looking at the intercalation reaction in lithium perchlorate and propylene carbonate. So each of these CVs were recorded in this small drop that was landed on the gold surface where the LFP particles were dispersed. And in this case, we're just changing the scan rate, but you can clearly see that we can monitor the intercalation reaction that's occurring. And the nice thing about this technique is that there is a residual solvent trace that is left once the meniscus is removed from the surface. And at the end of the experiment, it's entirely possible to take your sample and take it to an electron microscope, find the exact location where this measurement was made, and perform EDX, chemical analysis on the exact surfaces, or count the number of particles that were within the wetted area. One of the reasons why we ended up choosing voltammetry rather than galvanostatic type of measurements is because we are going to acquire an enormous amount of data, and it's much easier to automate data treatment algorithms using peaks. And so with the CVs, it's easy for us to be able to automatically extract the peak, the potential of the forward peak or the current of the forward peak. And it's also possible to carry out and extract the charge using the CV. So here on the left-hand side, you have a high-resolution stitched SEM image where LFP particles, which are the whiter aggregates, are dispersed. Each of the areas where we landed the micro droplet is highlighted by the circle, and the color of the circle in this case is the current of the forward peak for the intercalation reaction. You can then get really nice statistics. You can see that like a larger cross-sectional area correlates to higher peak current and increased charge, but you can generate a lot of data and evaluate a large number of battery particles using this method. In this case, one thing that we were able to provide in terms of information is how for a certain battery LFP batch, 67% of the battery particles that we had measured had an ideal electrochemical behavior. 27% and 6% had an altered electrochemical behavior, which basically meant that not all battery particles in this batch were created equally. And when we carried out further analysis using electron microscopy, it was revealed that for these particular battery particles, in fact, the carbon coating on the LFP particle wasn't uniform, and the ones that had the double peak and the shifted peak were not coated properly. So this is the summary for the story related to the batteries, where you can see that you can probe the localized electroactivity of battery active materials. Here I gave you an example for LFP, but we've also done NMC. You can change the organic electrolyte from propylene carbonate to a mixture or even to ionic liquids. And you can basically study a large volume of battery particles. Additionally, you can think about using SECM and an SECM instrument can also be used as a scanning ion conductance measurement, where you're basically looking at the changes in resistance. And if you choose to do this type of measurement, information that you can gain from it can be related to for example, the porosity of a cathode. So here you have a porous LFP cathode that was produced in a normal way for coin cells. And you can see that by carrying out similar type of approach curves as we discussed previously for the cancer cells, but in this case, we're looking at the ionic conductance in solution. We can extract the conductivity of the film and compare it to that in solution. So I'd say in recent years, more people have been trying to look at porous anodes and cathodes, whereas all the previous substrates that we had discussed were pretty solid. They had almost no porosity. All right, so now switching gear for my third story, it's to demonstrate how SECM can be used in terms of in the corrosion field. Now, if you've ever worked in the corrosion field, you know that alloys cannot be considered as a uniform substrates, just like a piece of platinum or gold. In fact, they have a lot of inclusions, which end up being corrosion inhibitors. Here you have a stainless steel 444 in the top panel that has these cubic inclusions. And these cubic inclusions ended up being the corrosion initiation site in this type of alloy. And industrial companies are very interested in knowing, well, how detrimental are they? And SECM can be quite good at providing a number. So in panel A, because the inclusion was cubic, we ended up developing a finite element method modeling for this. On panel B, you see you have the microelectrode. And at the bottom, you have the inclusion in light gray that's embedded in the dark passive film. And the rate at which the electron transfer is going to occur at the inclusion site or the passive film is not going to be the same. And so we should be able to observe this contrast by carrying out approach curves. So in panel C, we have our normalized current that's coming from the current of our mediator and our normalized distance. And you see the green and the red curves are the positive and negative feedback, the highest possible contrast that we could get. And the intermediate curves end up being the passive film and the approach curve recorded on top of a inclusion. And by fitting the approach curve to the numerical models, we can actually extract the heterogeneous kinetics for the inclusion versus the passive film. And one thing that's really interesting to see in the purple line is that the passive film, in fact, is not totally insulating. There is some reactivity because of the porosity of that passive film that leads to the regeneration of the mediator. So that's a nice example about how SECM can be used to study a broad range of corrosion alloys and focus on the impact of the inclusions in these alloys. Now, another industrial process related to alloys involves HVOF thermal spray coatings. This is when an industrial wants to protect an alloy further by forming a dense stainless steel, for example, film. In this case, you have an HVOF gun that combines oxygen and kerosene, and you introduce a powder carrier gas, which upon heating melts the particles and then forms really, really dense splats onto the surface that you're trying to protect. And in fact, this is a really large industrial process that is used in aerospace, aeronautics, and yet there are corrosion problems that exist with these coatings. If you look at here, you have a polarization curve. The black line is for the base stainless steel sheet. Okay? And you see that when you have the HVOF coated surface, the corrosion behavior is awful compared to the base material, even though the powder that was used was supposedly equivalent in nature. So SECM can be used in a similar way as I just presented before, carrying out approach curves on the coated samples. In green, you see the HVOF coating response versus the bulk passive film, which is in the solid red line. And clearly, the HVOF coated substrate is much more reactive. So just from a quick experiment, this type of experiment can take like 2 minutes to acquire to rapidly know whether or not the coating is doing its job and has a similar corrosion behavior to that of the bulk metal. In the end, we had to analyze single particles that were used in the HVOF process using the same type of methodology that I described for the batteries, where we just approached a pipette and carried out local PDP curves. And you can see in red in the D panel that in fact the source of the problem was not the coating itself, but the generation of the powder that inherently did not have the same corrosion resistance as the base metal. And so this type of story highlights that you can use the same instrument at many different scales, right? Like small stainless steel particle scale all the way up to big coatings. And the last example that I'll give you is a method that we recently developed, which is called the oil immersed scanning micropipette contact method. Now, one of the problems in having these small droplets is that you can have evaporation. And when you want to study alloys, it could take a long time to acquire the data. So it's really important that the droplet remains stable over hours, especially if you want to scan a large surface. And one thing that we realized is that if we cover the surface with a thin layer of oil, we get a much better performance in terms of background noise. So in the black trace, you see the noise was on average 2.2 picoamps in air. And if you had to humidify the cell in order to remove evaporation problems, the noise was really significant. But under the mineral oil, you had only 1.35 picoamps of noise. And so covering the corroding surface that you're trying to measure with a thin layer of oil really is going to help you get better signal to noise. And what's interesting about this method is that it really can perform many measurements over a large scale. Here you have an aluminum alloy, which we imaged. I think it had something like 1,000 landings. Each of the dark area circles on the upper left-hand side is a region where a droplet landed and measured either the corrosion current or a potential dynamic corrosion curve. You can see that the dark inclusions on the aluminum alloys actually show different corrosion potential on the top right-hand corner map. The areas in yellow have a very different corrosion potential compared to the rest of the aluminum matrix. And so this type of measurement can rapidly identify what feature is going to lead to corrosion initiation or be subject to microgalvanic coupling, which is also detrimental. If you zoom in, you can see too the our ability, like in 608, that's the yellow line, and 610 is the metal matrix. And you can see that the iron-rich inclusions, which are in yellow, exhibit a cathodic behavior relative to the aluminum matrix. And so what that tells you in a kind of measurement like this is that the surrounding aluminum matrix is going to be consumed as the anode, and there is going to be a galvanic couple with the inclusion. It's also possible to carry out a very similar type of map and then measure the corrosion current, for example, locally. And these experiments take a very long time, several hours, and yet the droplet is completely stable simply by putting a small oil layer on top of the alloy prior to the measurement. Here you can see another example where we carried out complete potentiodynamic curves on several areas. 29 and 12 are the aluminum matrix, and 30 and 13 are the actual inclusions. And so it's possible, based on these measurements, to identify which part of your alloy is going to be more susceptible to corrosion. So the conclusion is that the oil immersed version of SMCM is really nice because it allows you to use high evaporative electrolytes, like in the pipette for the measurements I just showed. It was 3.5 weight% NaCl, which is the typical standard in a corrosion measurement. And so using these high evaporative electrolyte solutions without the oil would lead to droplet evaporation normally and crystallization at the end of the pipette. But because of the oil, we can avoid this. And it allows for really long time stability maps. And so we really like this new application. And if you're interested in corrosion and you need to scan large areas, it's a really viable option. So with this, I will finish again with acknowledgements for the team that has worked on these studies. I wish we'd all be sitting in a room, but we can't. So thank you so much for actually attending the webinar. If you have questions, please put them in the Q&A section, and I will make sure to try to answer it to the best of my abilities. And final thoughts, today I talked to you about corrosion, cancer cells, and batteries, but SECM can also be used in spectroelectrochemistry, catalysis, biofilm, enzyme films, polymer films. It's a really versatile method. And it's also easy to modify the instruments to evolve over time. So thank you very much. And thank you to all the funding agencies that have made a lot of the work possible that I presented today. Thank you.
And thank you, Janine, for a really fantastic presentation. And yes, we will move on to the Q&A session. So the first question here, what are the similarities to SVET? So I think that's scanning vibrating electrode technique.
Yes, so SVET measures first of all, it's oscillating, right? And it also measures the difference. It sometimes has two wires. And SVET is also very useful in corrosion. So I would say it's like a complementary technique. Like most SCCM instruments could be adapted to do SVET as well. It wouldn't be that difficult. I'd say that the SVET probes are less developed than SECM probes at the moment. They tend to be much bigger. Hundreds, the entire tip is usually around 100 micron. So that means your lateral resolution won't be that good. So I would say maybe you can't do the inclusion, but if your objective was, for example, to measure galvanic corrosion between two dissimilar metals, so that could be quite good.
Excellent. Thanks, Janine. Um, Next question here. How can I measure the behavior of lithium batteries, uh, specifically electrochemical reactions using SECM?
So yeah, you can, for example, use the second story that I told was related to monitoring the intercalation reaction, but you can also use SCCM to measure the porosity of the anode and evaluate um, how um, wha- how much the mass transport is limiting in the cathode. So I would say for you can also I think there's also a nice group in Germany that used SCCM to monitor dendrite growth at lithium electrodes, and so I'll have your question, and I can suggest a couple of papers specifically for battery materials if you're interested.
Excellent. Thanks, Janine. Next question here from Daria, who's asked, is it possible to image the concentration of the ions involved in corrosion and to distinguish between their oxidation states needed for the corrosion mechanism?
Yeah, could you read it one more time to make sure I'm answering the question correctly?
Sure. So she'd like to know if it's possible to image the concentration of the ions involved in corrosion and to distinguish between their oxidation states necessary for the corrosion mechanism.
Okay, so thank you very much for the question. In our group, we've developed some magnesium selective ion selective electrodes. So when people want to detect a particular ion like magnesium or aluminum 3 plus, um, they'll make an ion selective electrode, and that electrode is sensitive to the activity of the ion in solution. And so there are reports on this. Now, in terms of the oxidation state, usually ion selective electrodes are based on a principle like they'll use crown ethers or molecules that will capture the ion. And so I apologize if the ion changes charge, it may need another molecule to capture it. And so, unless you can directly electrochemically react the ion at the microelectrode surface, you could plate it as well. If but and so other options too is to use micropipettes in combination with ICP OES, inductively coupled optical emission spectrometry. And so there, it's not SCCM per se, but you're collecting the solution and sending it to the ICP afterwards. So it's a complicated question. Um, and it requires the development of a specialized probe for you to do it.
Excellent. Great answer. Question here from Daniel, who said, thank you, Janine, for the nice presentation. Have you seen any effect of agglomerated LFP particles versus individual particles from the droplet measurements?
Well, so for sure, there is a difference, and for NMC particles that tend to have larger aggregates, yes, you don't get the same response. Like the current will be different.
Excellent. Next question here. Um, Nagash has said, hello, Professor Mauzeroll. That was a very interesting talk. I'm new to the field and would like to know if SECM can be used or has the potential for wet etching nanofabrication in situ using scanning imaging techniques. Do you or your colleagues or collaborators have any similar experience in this field?
Thank you very well. Thank you for attending and thank you for asking a question. Yes, etching surfaces and seeing whether or not the etched surface or in some cases, you know, in microfabrication you also have like copper deposition. SCCM has been used in this field. There are a few papers that I'm aware of, and in the Q&A answers that I'll do tomorrow I can provide some reference ideas, but definitely it's possible to use it.
Excellent. Thanks, Janine. And I think in the interest of time, we'll just ask one more question here. And yes, if there are a bunch of great questions that we haven't had time to address today, but we will be creating a Q&A report that addresses some of them, and we'll be distributing it to the full list of registrants for the webinar. So if you still have a question, it's not too late. Feel free to submit it now, and we will pass them all along to Janine for the Q&A report. So last question of the day will be, if we want to use SECM using the SGTC mode, how do we know or distinguish the current that we got is the current from SGTC instead of the current from feedback or bulk diffusion?
Well, so you have it's the key in substrate generation tip collection mode is to choose your redox reporter molecule really carefully. For example, and make sure that they're not in the same potential regime where they could contribute current. So it's when you start the design of your experiment, let's say you're going to carry out a reduction um, of oxygen and you're using a reporter molecule, well, maybe hexaamine ruthenium chloride isn't the best choice because it could like overlap with it. And so it's important to choose your redox indicators correctly because in substrate generation tip collection mode, you'll still want to approach the surface pretty close before you carry out the measurement. You could do this and also change the solution if you want it as well. That takes more time. So there's lots of strategies by which you can do this. And so it takes a little bit of experimental design at the beginning, asking yourself, what's the standard potential of the mediators? Will there be interference? So you do have to know a little bit about your substrate before you start imaging it.
Perfect. Well, thanks so much, Janine, for the really fantastic presentation, also for answering some of the questions with us today. It's been a real pleasure having you with us.
Yeah, and I hope all of you are safe. Thank you very much for coming.
Yeah, definitely. A big thanks to the audience for joining us today. And I'd also like to give a big thanks to Harvard Bioscience for sponsoring this event and making it all possible. So in closing, thanks again for taking part in this Inside Scientific webinar, and we look forward to having you with us again soon. Have a wonderful day, everyone.